So today we are going to talk about fungi. So the fungi, it is a term which comes from Latin fungus, which means that mushroom. It includes like they are eukaryotic spore bearing organism with an absorbed nutrition, and they doesn't have a chlorophyll. They mostly reproduce by means of your sexual and asexual reproduction. The scientists who study fungi are called as mycologists, and the scientists with dealing with fungi are called as, or the science stream which deals with fungi is called as mycology, and your fungi toxin. The fungal toxin study is called as mycotoxicology, and the disease caused by fungi in animals are known as mycococcus. Then comes your its distribution. So the primarily they distribute in their they act as a terrestrial organism, although they few are freshwater or marine. Some there are some which are pathogenic and infective in plants and animals. They also have a beneficial relationship with other organisms. For example, your lichens, which are associated with your algae or your cyanobacteria. Then comes your nutrition and metabolism, where mostly your fungi grows best at the most they grow in dark or moisture habitat. They also found wherever your organic component is more available, as they are saprophytes. They take up the nutrients from your dead and organic matters, and they absorb their soluble products also. Along with this, they are chemoorganoheterotrophs, which uses organic components as a source of carbon and electrons and energy source. Along with this, a glycan, which act as a primary storage for their utilized or taken up polysaccharide in the fungi. Mostly they are aerobic. Some yeast are facultative anaerobes. They obtain their energy by means of fermentation, like production of your ethyl alcohol from glucose, etc. Along with that, some fungi are obligate anaerobes, which mostly found in your rumen of your cattle. Then comes your I talked about toxins. So your Aspergillus flavus produces an toxin called as aflatoxin, which causes a disease called as aflatoxicosis, and they cause mostly in your rice, corn, sorghum, cereals, peanuts, etc. And along with this, they also called as ergotism, which causes by an mycotoxin called as ergot alkaloids, which is produced by your fungus Clavis purpura. Then comes your reproduction. So the reproduction, the image given here is your asexual spores, where first one is your transverse fusion. Then comes your fragmentation fungi or fragmentation hyphae, where your hyphae gets break down into pieces. Each becomes themselves as a spore. Then comes your chlamydospore. Chlamydospores is a thick-walled resting spores formed inside your hyphae. They are resistant because during unfavorable condition they make sure that the spores are safe enough until a favorable condition is obtained. Here example is Candida albicans. Then comes your sporangiospore or sporangiospores. They are non-motile spores formed inside a sac-like structure called a sporangium, and the stalk which holds up the sporangium is called a sporangiospore. So during favorable condition, the sporangium gets ruptured and the sporangiospores gets released. Next comes your conidiospore, produced externally, and it is a specialized structure called as conidiospore. And the organism which the stalk like structure which holds this conidiospores are called as conidiophore. Example, your Aspergillus penicillium, etc. Next, your blastoconidia, where your vegetative mother cell is attached or gets attached to a bud shaped enlargements called as blastospores, commonly seen in yeast. When favorable condition, these spores gets ruptured or not even ruptured, they get dispersed, and each cell gets to form as a new individual. Next comes your classification of fungi. So the classification of fungi, the first division is the division Zygomycota, then division Ascomycota, then division Basidiomycota and Duromycota. Along with this, and it also divides to a slime mold or watery mold. So in this slime mold and watery mold, there are three types. Division Myxomycota, Asciomycota and Oomycota. Your Myxomycota are called as acellular slime mold, and your Ascomycota is a cellular slime mold. In this presentation, we are going to talk about the first four divisions. The first division is your zygomycota. The fungi present in this zygomycota are called as zygomycetes. Mostly they live in your decaying plants and animals. They are saprophytes, but some are parasites for their plant, insects, and animals, even for humans. There's The hyphae are cenocytic, which is a multicellular cell or a hyphae, a cell or a hyphae without a septa. To When there is no septa, uh, there will be a rapid transportation of your nutrients, organelles inside your hyphae. This makes them viable during unfavorable condition. So as I said, they both are done asexually, and sexual reproduction is carried out here, where your asexual spores are dispersed in the wind, develops to a sporangia at the tips, like the aerial hyphae. At the part of the aerial hyphae, they forms the sporangia, where in your sexual reproduction they produces a tough thick walled zygotes called as zygospore that remains dormant when the environment is harsh for the growth of fungi. When favorable condition obtained, they get matured. So examples like your Rhizopus stoloniferan, which is a bread mold, along with this mucor. Here we're going to see about a mucor species. So mucor species, their habitat is mostly like an organic soil, a dead decaying matter of fruits, vegetables, and plants. And distribution, when it comes to distribution, they are throughout the soil. Wherever there is an organic matter content is more, they grow more. So then comes your reproduction. In terms of your reproduction, there are three stages: vegetative reproduction, asexual reproduction, then your sexual reproduction. Your vegetative reproduction is occurred by means of your fragmentation, where your vegetative cell gets fragmented during an unfavorable condition. After once a favorable condition is obtained, each fragments gets developed into a new vegetative body. Then comes your asexual reproduction. Here there are three. By three methods they can get reproduced. One is your sporangiospores, then your chlamydospores, and your oodiospores. First, your sporangiospores. So sporangiospores first, your vegetative hyphae comes. So this vegetative hyphae is formed from your germ tube. So these are filamentous structure. From this filamentous structure, they are called as mycelium, the network of mycelium. So from there, your conidiophore is produced. Conidiophore is actually a stalk-like structure where your sporangium gets produced or developed. So this is a specialized aerial hyphae grown upward from the vegetative hyphae. These are stalk-like structures that bears your sporangium. From there, your conidiospore gets matured. Conidiospore mature means you're at the tip of your conidiospore, there will be enlargement. This enlargement or enlarged structure is called as your sporangium or a spore sac. Inside your sporangium, which your cytoplasm gets accumulated and undergoes your mitotic division. From there, your sporangium, the actual sac-like structure, gets developed or differentiated. So differentiation, where your spores gets differentiated and they get developed and divided by means of mitotic division. A specialized structure called as colamen, which is present at the center of your sporangium, that makes them stable or connected with your sporangiospore structure. Once it gets matured, there will be this happens until a unfavorable condition occurs. Once a favorable condition is obtained, this sporangium gets ruptured and the cells gets, or the sporangiospores get dispersed in the environment. From there, once they reaches to a favorable environment, this spores gets to generate a germ tube. From this germ tube, again a vegetative hyphae is produced and the cycle is repeated. So this is your asexual reproduction of through sporangiospores. Next come your chlamydospores. So as I said, your chlamydospores are restricted thick walled structure. So first your formation of chlamydospore is done, and your from there maturation occurs. Once matured chlamydospores are obtained during a favorable condition, this chlamydospores gets detached and your germ tube is formed. From this, a new individual is produced. Next is your oodiosporium. Same as that your chlamydospores, here also your oodospores formed, then gets detached in a favorable condition and your germ tube is formed, and through those germ tube a new individual is produced. Next comes your sexual reproduction, where, as the name tells, sexual reproduction, it requires two different strains. So the two different strains are called as heterothalys. Mucor is, here we saw about the example of mucor. So your mucor is an heterothallic structure or heterothallic organism. This means that they require two different strains or a compatible mating strains, like a positive or negative. During this, once they get fused, they form a outward structure called as protuberance. So once this protuberance is formed, a small enlarged structure at the tips, center tips, this leads to a formation of progametangia, um, it is a multinucleated structure formed through your protuberance. This contains many nuclei, which this progametalia structure contains many nuclei. Upon this, a fusion of your progametalia that forms a conidio coenogametes. Coenogametes. This coenogametes contains many nuclei, upon which a zygospore is formed, where this wall between the coenogametes gets diffused or dissolved. This results in the formation of a young zygote. They remain dormant for a shorter time. During this dormant stage, all your protoplasm, like your plasmogamy and your karyogamy, occurs. This results into a formation of a thick walled structure, a zygospore. They are resistant, diploid structure. They are diploid structure. From there, your zygospore, maturation of zygospore occurs, where your under a harsh condition, they mature and remain as a zygospore for longer duration. From there, when the favorable condition is obtained, they produces a germ tube, they produce germ tube called as promycelium. At the tip, they contain a germ spore, which contains both positive and negative strains. When they get released, according to the spores, new mycelia, either a positive or negative strain is produced. Next comes your ascomycota.